I raised and born in Iran, and basically my passion to the medical imaging as well as using AI for good, it takes me to go around the world, starting from Sweden, where I did my master in biomedical engineering at Chalmers University. Then after that, I moved to Spain. I worked there for a couple of years in different area of computer vision, medical imaging, etc., etc. And after that, I decided to do my PhD. So I take the ferry. I move from one part of the Mediterranean Sea to the other part, from Barcelona to Genova, where I did my PhD in computer vision at Istituto Italiano Tecnologia, affiliated by University of Genova. During my PhD time, I had the pleasure to go to Chinese University of Hong Kong, where I did my internship there. And after that, I again reconnect to bring toward my main ambition to use AI for good. So I joined a multinational pharmaceutical company named Bayer. For those who do not know about Bayer, it is a company that creates aspirin. So I guess all of us know about aspirin. So I started my journey there as a postdoc research scientist up to today that I'm director of medical imaging and AI. Apart from my professional life, I'm interested in mountain biking. Specifically, I'm interested in downhills because it's just a moment that I do not think about work or algorithm, but mainly about myself and enjoying the trail. Apart from that, I'm interested in different languages. I speak Persian, English, Italian, Spanish, German. The life is too short to learn German, I guess, so my priority has been changed. And that's mainly about your speaker today. So the topic of today talk would be about synthetic data in medical imaging. But what I want to tell here is that this talk is not specifically about the technology, but rather about how we use technology for good. So it is not going to be a technological heavy presentation, but rather showing the impact that the algorithms and in the area that you're working, how it could really impact in saving lives. So, well, to do so, I would like to introduce you one of the most important contributor to humanity. It weighs only 25 grams, fits in palm of your hands, and yet has helped save millions of human lives. The laboratory mice, they play a central role in helping us to understand the disease, to understand the cure that we have for disease. So basically, they are the first main contact point that once we are coming up with a new treatment, we are testing on them to understand the efficacy of a solution that we have for a specific type of a disease, as well as the safety of them for before clinical development. However, the major problem of that is that we need to take lives to save lives. In today's meeting, I would like to show you how AI and synthetic medical imaging has the potential to reduce reliance on animal testing and accelerating drug developments. However, I need to set the stage before going to that to tell you what synthetic data is at all. Synthetic data is the data that is generated artificially without human intervention or physical or real-world physical interactions, and interestingly, is widely embedded in a seamless manner in our daily lives. Whether it is in entertainment, communication, content creation, is all around us. So, for example, a Hollywood movie named here, they use generative AI to synthetically de-age Tom Hanks. Many LinkedIn posts or social media posts, they are fully generated by generative AI tools synthetically, or they have kind of touch. It means that some refinement that has happened using those type of tools that are generated synthetically. Once we look at the computer vision area, the art industry is a dominant area that has been impacted. We are also able to do a lot of interesting, funny things with the generative AI tool to create synthetic faces, as you can see here, that what has happened for Michael Jackson's. So now the question here is that, can we use this technology to generate synthetic medical imaging? Now you may wonder, why synthetic medical imaging? It is because it is the most important data modality which is widely used by healthcare professionals to make important decisions about the status of patients, as well as understanding a disease. They are acting as a kind of window beyond our skins, and it helps the healthcare professional to look at what exactly matters to them. When we are looking at the drug development part, specifically on a preclinical setup, you could see two images of a mice, that once you inject the medicine on the mice, you want to see that what is the concentrations of the drug on a different part of the organs for a different reason. So that only is possible by using non-invasive approach or semi-invasive approach, because some of them are invasive, which are medical imaging. So with the rise of AI, as well as importance of the medical imaging, it creates a significant potential to exploit the market opportunity. It's estimated that the market size, it will increase from $1.1 billion in 2023 up to $40 billion by 2030. However, the major challenge that we have to leverage this market opportunity, as well as using AI for efficiency that we want to have in drug developments, there is a significant barrier to that. We are often cases, the data that we have available in the healthcare domain, they are suffering from data scarcity, which means that the data, we have a limited access to the data with a high quality and diverse quality that hinder the drug development. And it's slowing the progress and increasing cost for us, as well as it developing any kind of AI algorithm would be difficult for us. Sometimes we are dealing with data imbalance, which means that we have an uneven distribution of available data. For example, in the case of rare diseases, where we have only few samples from real world scenarios, that unfortunately it creates data imbalance that not only our decision, but also whatever algorithm that we want to develop for supporting healthcare professionals, it would be biased to the specific populations. Sometimes we have a data, but unfortunately the data are fall behind the firewall of regulatory and privacy concern that we have, and we are not able to share the data. Therefore, there is a limited access to critical information for research purpose. And sometimes, ethical consideration, as I mentioned before, we need to take the lives to save the lives. So it means that we only, to create the real world data, we need to kill mice to be able to generate real world data. So I give you a kind of introductions that data scarcity is a major challenge, but I thought that would be also a good idea to provide you a practical example what it means data scarcity at pharmaceutical company. So we had a use case that basically we were aiming to provide the AI tool as a kind of software as a service on the existing drug that we have in the market, that basically promote and help to increase the sale of this specific drug at the markets. The the modality that we had, it was on MRI images. So we decided to kick off the project. So we immediately went to acquire data. We acquired 7,000 patient data with a cost of $1.5 million dollar or euro. I cannot be sure if the currency it has been euro or dollar. However, we as you can see here, during quality control, we have like such a data heterogeneity, data imbalance, and some data was really low quality. Only 2,000 of them was usable for training AI model. It means that 80% of those data, they did not meet our standard. So to highlight that, it means that 1.2 million euro here, it has been wasted due to the low quality data. And this cost is excluding the cost of data science, data engineer, radiologists, and also cloud computing. So it means that if we would have had known this before, we would have been able to reduce significant costs that we have created. How you may wonder that synthetic data would have been able to help us. We could have, if we would have access to synthetic data, we could have changed our larger scale investments. So it means that we, rather than defining a problem and looking for vendors and acquire data with a huge cost and then train the model, see that what would happen, we would have maybe been able to do the incremental approaches. So basically starting with the lower scale, having generator that or foundational model that is able to generate synthetic data, we add it. If it works, we train the model. We see that if it works, then okay, let's invest more. If it doesn't work, then stop. And we continue this loop further and further and see that if it works, then we add. If it doesn't work, we stop. So in this approach, we would have been able to save some kind of cost, but unfortunately, we learned our lesson in a very expensive way. So so far, I gave you an introduction about what synthetic data is, why medical imaging, and the importance of medical imaging for a pharmaceutical company, as well as for diagnosis and prognosis of a disease. After that, I moved toward actual real world challenge that we had in our company and how synthetic data could help. Now, from now on, let's go back to our actual use case, that how synthetic data is able to reduce the number of sacrificing of a mice. So the major challenge, as I mentioned, that I mentioned before, is that the process is time consuming. We have also tremendous pressure from authority to reduce our animal testing, but remember that we are not sacrificing those animals for fun. We testing our hypothesis on them to understand that the solution which goes to the patients are safe. And if we reduce the number of animal testing, it means that our real world evidence data would reduce. So we have less certainty about the decision that we have about the status of our drugs. So now the question is that if you are using, how we could use synthetic data to reduce animal testing, while at the same time we ensure that we are able to keep the same standard as we are killing or sacrificing animals. So let's go over the preclinical drug development testing, from injection to digital pathology. So we come up with it's a very rough, like a presentation. It means that we come up with the new drugs, we test it on the animals, and then we sacrifice them, which we call it necropsy. Then we extract the different organs from the mice, and we look at, for example, there are key organs We look at the brain because we want to see that what is the impact of a drug on the nervous system, what is the impact of a drug on the respiratory system, for example, on the lungs, what is the impact of a toxicity of a drug by looking at the liver and the kidney of the mice. Then once we extract the tissue, we have a kind of a process which we call a digitalization, and also a staining. A staining, it means that we add a color to make the tissue visible, and whole sliding imaging, it means that we need to create a high resolution, 20 to 40 times bigger than the actual size to make them more visible. And after that, we send it to the pathologist, and they're looking at those images, and they understand the tumor response, drug induced toxicity, inflammation, and immune response. So as I mentioned, this is the current workflow, and what we want to do now is that we would like to use a generative AI to reduce the animal testing. So it means that our approach is only focusing on the historical real data. We use that data to train a generative AI, and after that, once the model gets trained, it generates us synthetic imaging, and from there we could send those synthetic data to histopathologists, to pathologists to be able to study about the effect of potential drugs. So now you may wonder how we are doing that. So the structure of a generative AI approaches that we are using is not based on GANs or variational autoencoder. We are mostly focusing on the diffusion model, and particularly a stable diffusion model, because training is less expensive for us. So the internal mechanism of a diffusion model is as follows. So we have an original image, and it consists of forward diffusion and reverse diffusion. In the forward diffusions, what we are doing is that we are adding incrementally noise on top of the original image, such that at some point in time it gets fully cropped by noise. And in a reverse diffusion, what we are doing, we are reducing a noise by denoising the image from fully cropped one to the original images, which we call it, like a reconstructed image. Or here in this image, I name it as a generated image. So the ideal output should be as close as original images. But once we are training such a model, we need to also have a kind of control on top of what we want that model generate for us. So every images, every data that we feed into the stable diffusion models, it should be multimodal. It means that image accompanied by text descriptions or associated text. So we have images, and we have associated text here, for example, which describe the content of the image. So in this case, for example, we have a bulldog walking on the grass with a wooden object in a mouth. So it turns out if you repeat this process with the sufficient, the more data you have, the better it is. With the sufficient amount of data, the model is able to generate for you the high quality images. So once we train the model, then we need to generate new images. So during inference time, what we are doing is that we need to only provide as a prior Gaussian noise plus the descriptions of the image that we want that this model generates for us. For example, in this case, I would write dog with wooden object in the mouth, and it creates several examples. Some of them are good, some of them are not good. That synthetic images of a dog with a wooden object in a mouth here. So we want to use the same technology to generate histopathology image for us. So we use the same model to do it to do so for us. And here is the initial result that we get. And you could see here is that our model is able to generate synthetic histopathology images, but they are looking very much as like artistic. And then, for me, honestly speaking, it has been a very good sign, because the model was not having any kind of experience on generating synthetic histopathology images, but still it's able to read between the lines and generate some artistic looking images based on the information, based on the best knowledge that it has. So we use those images for fun, for a backup of our some team meeting that we had, but obviously they are not usable for diagnosis or prognosis purposes. However, this result has motivated us to look more closer to the use case. So and we train our stable diffusion models for these specific tasks that we are looking for. So we start with whole slice images. As I said, whole slice images, they are gigantic images. They're super big images that they are not, with the current computational power that we have, we are not able to process them all at once. So we need to extract the different patches from them. And basically we create, for each image, maybe we would have, 5,000 patches. Depend how you patch, is overlap, non-overlapping, etc., etc. And then for each patch, we should have kind of medical reports. And using these informations, we should be able to train our diffusion model the same method as we train the wooden objects of the bulldog with the wooden object in the mouse. So in this case, we only replace the image information with the histopathology images and textual information with the information that we have from our medical report. However, a major challenge that we have here is that medical report often does not exist. And that is the process that we conduct most of our research based on that. Yeah. So we start to do our research in these directions, and what we have done here is that we have whole slice images. We had very limited information about the type of the prompt or information that we have about the patches. Either they have been cancer or they have been healthy. Then we build a prompt out of that. So histopathology image of a cancer tissue, histopathology image of a healthy tissue. So there are two classes, as I said, and we build a template-based prompt out of it. And then what we have done is that we train our diffusion model based on these prompt information that we have. Well, we can see that the result is not bad. It doesn't look artistic image as it was before. However, you can see that the real data that we have is much diverse compared to the synthetic data that we have generated. And then the reason is quite simple, because once the prompting is so high level that the generated images, it has been only focused on some specific region of real data that we have. So it means that the distribution of some type of a structure that we had inside the histopathology data that we had, it was very much prone to some specific features that the model it get comfortable with, and they get lazy, so they didn't do more exploration. So therefore, they repeated to create some specific patterns that it does not fully cover the real world evidence. So that has motivated us to move forward with this topic and define our own way of prompt generation, which we call it morphology-driven prompt clusterings. So what we have done, again, we have a whole slice image. We generated into the healthy and cancer one, we use a kind of feature extractor, which we call a dino model. People who have been working on deep learning, I guess they are familiar with this pre-trained model. You can think of it as a kind of feature extractor. And then what we have done, we created embedding a space based on a dino feature, and we apply morphological clustering, or k-means clustering, with the 33 cluster centers, and we create a new prompt. So a simple approach is that histopathology, image of a cancer tissue, morphological type 1, morphological type 23. For each class of cancer and healthy, we have generated such a prompting approach. And then we use this prompt to train our model. It's worth mentioning that the research has been also published as a journal publications, and the full report of our like a methodology is already there. So now you can see that doing this practice, we are able to generate synthetic data that is following closely to real world data that we have. And this is also obvious here. Here you could see that, the blue dots, they are the real world data distributions, and the black dot are synthetic data morphological space, or our proposed approach that you could see that is much diverse. It still is not perfect, however, is much better compared to what we had initially in a naive-based prompting approach. So what we have done, we have conducted extensive proof of concept study there, and what we have done so far is that we computationally and also visually assess our algorithm. As I mentioned, the full report exists in the paper that we have published. We recruit 5 expert pathologists, and we provide them with the 40 images. But unfortunately, or fortunately for us, they were not even able to distinguish between real and synthetic data images. And that shed the light for us that these approaches, they are like able to generate realistic looking images. However, I should also emphasize that the imaging data is one of the data modality that the subject matter experts are looking at. However, they are looking at a different aspect of one patient or mice to make their final decision. For example, the vital signs, their medical reports, the pathology that they have, etc., etc. These are also playing important role to their decision making. However, we have shown that we are able to generate high quality images that they could not distinguish between real or synthetic ones. However, as I said, we have also, like I thought it would be also fair to show that the challenge that we had. This is the situation that we have seen the model somehow get failed to be generalized. You could see that on top you have a real data, and below you have a synthetic ones. We observed that if you let the model to train more than some specific epochs, the model they get again comfortable with some specific feature, and they're able to generate, although colorful, but very similar structures. So this is very important consideration that we need to take into account while we are dealing with generating synthetic data. So it shows that the importance of a validation pipeline in place to be able to train these models and assess the output of those image generators before putting it into the practice. So, as I said, we are extensively working toward our vision to be able to generate to replace or reduce the animal testing. We as a company, we have had several collaborations to make that happen. One of the innovation, one of the most important activity that we are doing, you could also search for this program Victory R, that we are also developing, our colleagues, they are developing and implementing virtual control group to reduce animal testing, which they are used in a toxicology research. This is five years, I guess, research program with a significant funding, with a lot of partner across the universities, as well as corporation and companies in the pharmaceutical space to make that happen. And in general, synthetic data, it received a significant attention from authority, from authority perspective and funding agency within Europe, and there are several funding is going toward that directions. However, as I said, synthetic data is not just about the images. It is a complex world. It's a multidimensional ecosystem that integrates EHR, genomics, imaging, and even beyond that. So my personal dream would be, our objective would be to generate synthetic human. So it means that you would be able to generate, for example, 50 years old patient, smoker with lung cancer, put it into the kind of system generative AI, and as the output, it would be able to generate you electronic health record, genomics data, imaging data, etc., etc. This is a very active research area, and I see that we would have a lot of activity to do along this line. However, I thought that it should have been a good idea that to tell you about also pitfalls that we have. You could see that the pace that we see in a technology space is not, you do not see the same pace, same development pace that is translated to the pharma or drug discovery. There is a couple of reasons behind that I thought that would be interesting maybe for students and faculty know about that. It's a regulatory complexity. It means that every solution must meet the strict compliance, like a GXP, MHRA, FDA, EMA. And the innovation is constrained by safety and privacy and validation. It's very much regulated environment to act in. You cannot be really innovative. You really need to be careful about the outcome of the decision that you're making. Another problem is long timelines and slow iterations. For your information, drug discovery pipeline, it takes 10 years plus to basically conclude one drugs, and feedback loops are very long and expensive, which making agile development very tough. Another thing is that I should say that pharma world is not a technological world. They have their own proven success story even without AI and technology. So you could see that we have now data science at the company and also subject matter expert. There is a huge misalignment in priority and language between the data scientists and domain expert. As data scientists, we are very much excited about technology, but you could see that as a person with lots of years of experience at pharma company, you know how the business is working, and you're very much risk averse and process heavy. So I'm strongly believing that this cultural shift will happen, and we are under a process of transition. However, this is a major bottleneck, is to bring and translate technology to a pharma world. And the other thing, and last but not least, very important topic, is that in the drug development part, when we're talking about patient life, good enough doesn't work for us. Every result must be significantly explainable and reproducible, and you should remember that the output of the models is not benchmarking on a Kaggle or writing a paper. They are really a patient outcome, and it has a direct impact to the patient life. So I would say that, as a closing note, synthetic data is more than just a technical tool. It's a foundation for reimagining how we explore the biology, diagnosis diseases, and accelerating innovation. As I said, I only present right now on the imaging side. However, we have a protein foldings, we have compound discovery, etc., that they are also leveraging from in silico approaches, and they are very important topic that one needs to look at that. And we are looking at this as a community. As this field is growing, in my opinion, is that rather than having more smart and stronger model, the most important part is that we need to have a new talent, new thinking, an academic program that can reflect this shift. And I would be very much interested in collaborating with the university on mentoring a student and working alongside faculty member in evolving in these evolving fields. So if you have any requests on collaboration, questions, or just continuing conversation or get to know, please do not hesitate to contact me. My pleasure would be to hear about you and basically to continue a conversation with you. Thank you very much.